This is a protein, 10 amino acids in a row. But it never stays in a row. It folds. And the string is not the shape. That's the mistake that quietly wrecks this whole unit. The sequence is the instruction for the shape. Sequence sets shape, and shape sets the job. 3 minutes. Let's read a protein end to end. 2 amino acids join by squeezing out of water. What's left is a peptide bond, strong and covalent. 10 amino acids linked in order. 10 beads, 9 bonds. Remember that 9. This ordered list is the primary structure. One dimension, just the order. Building removes water. Breaking adds it back. But that bond itself? Hard to break. Now the backbone starts bonding to itself. Coil it up, and you get an alpha helix. Or lay it flat into a beta pleated sheet. Same idea, different fold. But look what's holding it. Hydrogen bonds. Weak ones between backbone atoms. Not the strong peptide bonds. And the side chains? Still just along for the ride. Their turn is next. Because now the whole thing collapses into a blob. This is tertiary structure. And what folds it is the side chains, finally reacting, pulling, bridging, clustering. One of them, the disulfide bridge, is even covalent. Covalent, but not primary. Hold that thought. The oily amino acids get shoved to the center, not because they attract, but because the water squeezes them there. So the R groups aren't decoration. They're the whole reason it has a shape at all. Some proteins go one step more. Several folded chains snapping together. Hemoglobin is 4 of them, 2 alpha, 2 beta, held by the same weak bonds just between chains. Pull it apart and you get 4 whole chains back. Not every protein does this. Many stop at 3. Here's the quiet miracle. That one sequence folds the same way every single time. Not random, not luck. The order decides the fold. Now crank up the heat. The shape unravels. It denatures. The job stops. But watch the peptide bonds. Still 9. They never broke. Heat only takes the weak bonds. A peptide bond is almost 20 times stronger. It survives. So denaturing loses the shape, but keeps the sequence. That's the whole definition. It's a fried egg. The white turns solid because the chains unfold and tangle, not because anything got chopped. And since the instructions survived, some proteins just fold right back. Now the payoff. Change 1 amino acid, just 1, from charged to oily. In hemoglobin, that single swap makes a sticky patch, and the molecules stack into stiff fibers. That's sickle cell. 1 amino acid out of 146. No bond broken, just a different molecule. 1 letter changed the shape, and the shape changed everything. This is why shape is everything. The fold makes a pocket, an active site, and the pocket does the work. Warp the fold, and the pocket's gone. Same amino acids, wrong shape, no reaction. So read it all the way down. Sequence to backbone bonds, to side chains, to the job. Sequence sets shape. Denaturing keeps all 9 bonds. And 1 amino acid can rewrite the whole thing. Structure determines function. Read the sequence and you can read the protein. Want to fold this one yourself? Links below.